With more than 6,000 small and micro-cap companies listed, if you're looking for the next apple at the earliest stage, then Channel Check truly is the orchard. The listed companies support Channel Check, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for Channel Check so you're always up to date on what's going on at the small and microcap data place. Welcome to this special edition of the C-Suite series presented by Channel Check and Noble Capital Markets. Noble is an SEC-registered FINRA licensed broker-dealer and a source of the equity research available on Channel Check. This investor forum at the World Stem Cell Summit presentation features GID BIO. Following a brief overview presentation from CEO William Cimino, Noble Biotech Research Representative Greg Arand will moderate a Q&A session. If you registered for this event through channelcheck.com, in the coming days you'll be given an opportunity to get your own questions answered by the company. With that, I am pleased to present Dr. William Cimino.
Thank you for joining today. My name is William Cimino. I am the CEO of GID BIO. I'd like to thank the World Stem Cell Summit and Channel Check for hosting this event. This presentation has forward-looking statements. We are raising a financing in form of a convertible note financing of $20 million. These convertible notes will have an automatic conversion on triggering events, including a qualified offering, acquisition, and majority approval of both the holders and the company. The conversion price will be the lesser of a 20% discount or a conversion cap price, which is $80 million, fully diluted enterprise value. Repayment of the notes will also include shares of common at the convertible cap price and 25% of the number of converted shares in the form of warrants. It is a 6% annual interest rate and the note is 3 years. GID BIO is a cellular therapy company currently in pivotal phase 3 clinical stage. Our technology enables point-of-care cellular therapy using a patient's own cells for treatment of significant degenerative diseases, including osteoarthritis, peripheral vascular disease, and Parkinson's disease. Our biologic cellular therapy approach leverages the most powerful healing mechanism known, the body's innate and natural ability to heal itself. Our device technology isolates and separates the healing stromal and vascular cells, including stromal progenitor cells, from the patient's own adipose tissue at point of care and treats in a single procedure. We have completed phase 1 and phase 2 efforts and launched an FDA pivotal phase 3 clinical trial for our technology and lead indication, which is osteoarthritis of the knee, the largest market in musculoskeletal disease. Our FDA Phase IIb study for osteoarthritis of the knee showed that 85% of subjects responded greater than placebo at 1 year, with a median 87% improvement in pain, stiffness, and function. We have a significant clinical pipeline for treatment of other degenerative diseases, with pilot human data and development plans for osteoarthritis of hands and spine, diabetic ulcers, and Parkinson's disease. This is our board of directors. Our chairman is Roy Bostock. He was previously chairman of Yahoo and chairman of Delta Airlines and Northwest Airlines and a director at Morgan Stanley. J. William Futrell, chief of plastic surgery at the University of Pittsburgh for 20 years and past president of the American Association of Plastic Surgeons. He is also a founder of the company. Morgan St. John, active in investment banking for over 40 years. Nick Pachuta, worldwide president of orthopedic innovations at J&J for 10 years. John Maris has led multiple development and exit opportunities in the medical technology space. And Adam Katz, currently a professor of plastic surgery at Wake Forest and a founder of the company, and internationally recognized for contributions and innovations in adipose tissue technologies. This is our management team, including myself with over 30 years of experience in medical product design, development, and release. Dale Tomerley with over 30 years experience in operations and finance. Ashley Wodashek with over 7 years experience in the FDA as a consumer safety agent, and Adam Katz, our Chief Medical Officer. Knee arthritis is an immense and growing market with unmet need. Early interventions are symptom modification, which is pain management. This is the realm of drugs with poor to moderate pain relief and improvement in function, generally having a very short duration of effectiveness between days and weeks, and side effects that limit reuse. On the other end of the spectrum, when disease becomes debilitating, we have major surgery, which is joint replacement surgery. These are devices used at the end stage of disease. This is a major surgery and recovery, significant rehabilitation, and possible serious complications. The benefits of this surgery are that the therapy has a durable result and tends to have moderate to good improvement in pain and function. The vast space between symptom modification and major surgery is an unmet need. This is really the space of disease modification and really the value of a cellular therapy, a biological approach to address a disease-modifying therapy, where we would look for moderate to good pain relief and improvement in function, a result that is durable, also that is minimally invasive and with minimal side effects. A few notes on the FDA and cellular therapy. The FDA regulates cellular therapies and cellular biologic products through CBER and the BLA or PMA approval process. All companies or providers must engage in this FDA process to legally market their technologies or process for a cellular therapy. GID BIO has worked with the FDA on osteoarthritis beginning in 2014. We received an FDA approval for our pivotal Phase III efforts in March of 2020, and we are currently enrolling and treating patients. The FDA 3-year discretionary enforcement period ended in May of 2021. Expect an increase in FDA enforcement activity for non-approved stem cell clinics and stem cell products and off-label use of non-approved biologics. There are 2 fundamental cellular therapy challenges. The first we call the product challenge, and that is to deliver a cellular therapy product with a process that is reliable, characterized, and quality controlled. These are FDA questions. Solutions are measured in terms of process controls and quality controls for production of the cellular therapy product. The therapeutic challenge is to demonstrate safe and effective clinical outcomes using the cellular therapy product to regenerate, repair, or replace diseased or degenerated cells and tissues. These, again, are FDA questions. Solutions are measured in terms of clinical outcomes and the relationship to types, quantities, and qualities of transplanted cells and the transplantation process. GID biotechnology addresses both of these challenges. The GID solution to the product challenge is point-of-care cellular isolation and transplantation. On your left, you see our patient and the 3 basic steps of the point-of-care cellular isolation and transplantation process, which are harvest, then process, and then treat using a single dose in a single procedure. Adipose tissue is harvested from the patient. Typically 30 to 50 cc's of adipose is harvested directly into the tissue processing device. The processing then takes about 60 minutes to generate the stromal vascular fraction cells, which are then analyzed for release and then used to treat the patient directly in the figure as shown, directly injected into the knee. On the right, you see a little more detail of this process. We start with whole adipose tissue is harvested in the small lipoaspirate harvest. That tissue is disaggregated, and then an isolation and separation process is used. After it takes about 60 minutes, generates more than 1 million stromal vascular cells per gram of tissue with an average viability of 80%. This is essentially reducing a GMP tissue processing facility to a single-use disposable device that is used at point of care. As a result of this processing, we isolate and retain the stromal and vascular cells, and we isolate and remove cells including the adipocytes, extracellular matrix, leukocytes, erythrocytes, the local anesthetics used in the harvest, and cellular debris. The therapeutic challenge is addressed with clinical trial results. Our Phase II randomized controlled study for osteoarthritis is an FDA-approved study under IDE. This was a multi-site, randomized, double-blinded study, placebo-controlled, with parallel groups, 3 groups, and efficacy follow-ups at 6 months and 1 year. This is a single injection of stromal and vascular fraction for the treatment of osteoarthritis at the knee. This was published in the Journal of Sports Medicine, American Journal of Sports Medicine. You see the reference here. This was published in March of 2020. In summary, we met all efficacy results. We successfully met all pre-specified efficacy endpoints. We had a strong safety profile, no serious adverse events, no adverse device events, and no unanticipated adverse device events. That was through a 2-year safety follow-up. This is a snapshot of the output of the trial, the results. What you see is a plot, and across the bottom you see time. This is 1 year, 52 weeks, and up the y-axis you see the total WOMAC score, which is the leading measurement for osteoarthritis pain and function, expressed as percent improvement. On this plot, up and to the right is improvement, and down and to the right is decreasing improvement. The green line that you see here is our high dose response over 1 year. This is the low dose response in gray, and this is the combined low plus high, the treatment group, in blue. What you can see for the high dose, for example, is over an 80% improvement at 6 months and maintained out to 1 year. The treatment group, the combined group, with an over 60% improvement at 6 months and over 80% maintained at 1 year. The orange curve is our placebo curve, which is declining after an initial rise down to about 25% at 6 months and decreasing back to 0 at 1 year. What the FDA is interested in is clearly the difference between the placebo curve and the treatment curves, both at 6 months and 1 year. In our case, we were statistically significant using a superiority test at both 6 months and 1 year. This is a comparison to historical data generated in published results for the alternative treatments for osteoarthritis. You can see our data, which is the green curve for high dose and the blue curve for treatment dose on the very same curve, 1 year, or the same plot, 1 year across the bottom, and the percentage change in total WOMAC up the y-axis. The hyaluronic acid injections and corticosteroid injections and their controls are shown in this area here. You can see that it largely represents the same value as the placebo in our studies. Roughly 20 to 30% improvement in 6 months, and no data thereafter because these are short-acting treatments. The total knee replacement curve is the gray curve you see here. This is about 50-55% improvement at 6 months, maintained out to 1 year, and clearly maintained beyond that. What you can see in this entire plot is the differences between our biologic response and the other treatment modalities that are available. This is a look at our development status and pipeline across the top. We have the stages, phase 1, phase 2, phase 3, and the approval stage, and then an estimated number of obtainable procedures should that get approved in the rightmost column. OA of the knee is our leading therapeutic. It is currently in phase 3, enrolling and treating. We are expanding that into osteoarthritis of the extremities, primarily the hands and also the spine. We have human data for hands, which I will discuss in just a second. Parkinson's disease is a neurodegenerative disease. We have initial human data and an ongoing pilot study for further human data. We have wound care at the bottom, which I will present a few results for diabetic foot ulcers. Let's start with the diabetic foot ulcers. We've conducted 2 pilot studies. This is a very visual presentation of the evidence of the healing power of stromal vascular cells. What you see in these pictures are 5 out of 50 patients that participated in the study. On the left, you see the preoperative result, and on the right, you see the results at 6 months. In each, over 94% of the patients that had diabetic ulcers that looked generally like this were fully closed at 6 months. 3 achieved 75% closure, and there were no non-responders. I should point out that there are no skin grafts and there are no sutures or no other closures used. This is a single treatment with stromal vascular cells and then results at 6 months. This is our data for hands. These are 10 severely arthritic hands. On the right, you can see an example of what these hands look like. This patient was unable to do a basic pinch and hold test or to write after treatment. She was able to both pinch and hold and to write legibly. This is published in Cellular 4. On the left, you see the graph, which is very similar to the osteoarthritis graph for the knee. This is 2 years across the bottom, and this is the percentage improvement up the y-axis. And you see a very similar response to the knee, which is an improvement out to 6 months, and then maintaining that response at over 80% improvement out to 2 years. This gray curve you see here is a strength curve. This is a dynamometer, and you can see that we maintained over a 40% improvement in strength out to 2 years. This is Parkinson's disease. This is a small study of 2 patients. There is an ongoing study with 10 more patients in Parkinson's disease. What we capture on the left is 2 segments from a video attempting to show how the patient responded pre-treatment in 1 year for a patient getting up and out of a chair. You can see their motor skills are greatly impaired on the left and much more able to rise out of the chair 1 year later after treatment. The graph on your right shows the Parkinson's disease questionnaire, which is a standard questionnaire for Parkinson's patients. And what you can see is a tremendous improvement in mobility here at 1 year and also a decrease in pain. This was published in the Journal of Clinical Neuroscience. This is our regulatory path for osteoarthritis of the knee. It would be similar for other indications. We have completed phase 1, phase 2 efforts here as shown here and published the results, and we've now been approved to move into the phase 3 efforts. We are approximately here in that effort. Our intellectual property platform is very strong. We have 10 issued patents in the United States, 4 issued in Europe, and 1 issued in Brazil. We have 9 pending in those areas. Our coverage is generally in 5 large patent families. These are apparatus and method claims with earliest priority dates between 2010 and 2017. The competition for knee OA is an immense market, totaling about $9 billion annually in the US for the technology phase. For the symptom modification, really pain management, we have drugs and injections occupy about 33% of that $9 billion annually in the form of 3 million steroid injections, 1 million HA injections, and then a large number of use of drugs, primarily NSAIDs and some opiates. On the other end, we have joint replacement surgery on the right side here, occupying about 66% of that market with over 900,000 annual procedures, 6 billion annually split between the large manufacturers of total joint replacement implants. In the middle, there is actually nothing that is a disease-modifying therapy available, and hence the value of a cellular therapy, an approach that can generate a disease-modifying approach and address that unmet need. Our strategy is to use the proceeds from this financing to complete our pivotal phase 3 effort and submit for a PMA, gain FDA approval for osteoarthritis of the knee, develop 3 pipeline efforts to the phase 2b randomized controlled level, and establish a reimbursement pathway for our approved treatment for osteoarthritis of the knee. We are poised to become the first FDA-approved cellular therapy for osteoarthritis, which is a cellular therapy in an immense market with an immense unmet need. Why GID BIO? We have a differentiated technology platform with strong patent protection. We have a strong pathway to commercial sales and reimbursement, and we've created multiple collaborations. We have a very lean operation without setbacks and a proven ability to engage with the FDA. We have a path to first to market for a tremendous unmet clinical need in a large and growing market. We have a strong clinical data platform and an experienced team. Thank you very much.
Hello, this is Greg Arand at Noble Capital Markets. Thank you, Bill, for joining us today and for their excellent presentation. I do have some questions about the information provided. I'll lead off about the actual adipose tissue that you use for your source of stromal and vascular cells. Why adipose tissue?
Adipose tissue really addresses one of the most important needs, and that is to be able to generate a therapeutic dose of cells, meaning a sufficient number of the right types of cells for the therapy. And adipose tissue is the richest source of stromal and vascular cells in the body. So it is a very high concentration of stromal vascular cells. And secondarily, as a tissue, it's available for harvest, meaning we can get enough of that tissue and with the high concentration to generate a therapeutic dose at point of care, thus eliminating the need to culture or expand the cellular harvest. So it's really enabling with regard to being able to generate a therapeutic dose.
I certainly have enough adipose tissue as a source, so I assume that's true for a lot of people. Are there actually stem cells in stromal vascular fraction, and what are stem cells exactly relative to utilizing from this type of tissue?
So the definition of stem cells is a sort of a co-opted term, but let's reduce it to a very simple structure. In adipose tissue, we have stromal progenitor cells. These are very nascent early cells that would become stem cells. They have stem cell properties. Stem cells are cells that have been propagated, usually in culture, and they have two primary characteristics. One, they can replicate. And two, they have some level of plasticity, that they can be differentiated across multiple sort of lineages. And by that, I mean different types of cells. So these cells are present in their nascent form, which we call stromal progenitor cells, in the stromal vascular fraction. They could then become stem cells if they're cultured, and if they're cultured out of adipose, they are called adipose-derived stem cells. If they're cultured out of some other tissue, they would be whatever that other tissue is, stem cells.
You talk about, prior to this, a therapeutic dose. Is there a limitation, and why is it important to consider therapeutic dose levels?
A therapeutic dose is critically important. And we have, for our studies, the FDA's required dose escalation studies, and the intent there is really that you show the difference between a placebo, for instance, would have no dose or no therapeutic dose, and then a medium, and then a higher dose. And the intent is to try to draw what's called a dose response curve. If you have insufficient numbers of cells for a therapeutic dose, you're not going to get an output. Once you achieve a therapeutic dose, you're going to get, hopefully, your targeted output. And so it is critical. So the important issue here is that it is not simply a sample of cells taken from somebody's body, uncounted, and you don't really know the dose that you are applying back to a patient, versus having a sufficient dose to generate a reliable and consistent therapeutic output. And that's why knowing your therapeutic dose and where those bounds are is so important, versus essentially a guess or an arbitrary amount of tissue or cells which are not quantified, and therefore no relation to a sort of a consistent and reliable therapy.
As a follow-up to your other comments about your Phase IIb studies in osteoarthritis, you had excellent significance statistically relative to placebo. Can you give us any details or insights into how Phase III is working?
I can't tell you what the results are of Phase III because I'm blinded, of course. I can tell you what the FDA is required in Phase III, and I think that that's important. The FDA, it's again a randomized, controlled, blinded, placebo-controlled study, parallel group design. The FDA is further required that we isolate and separate the pain and function scales as separate scales, so that we are evaluating these as independent tests, both pain and function, in order to complete the Phase III effort.
You had a long list of pipeline programs. Can you elaborate further on some additional programs, specifically in Parkinson's disease? That seems like a really hard to treat area, but also the ulcer program that you talked about.
Let's start with the ulcer program. That was really one of our first areas of investigation, because it's such a, you can see, it's such a graphic need and a terrible situation for patients. All those patients were amputation candidates, and that's really all that is available to them. And we were able to successfully treat them and actually close the wounds, and we now have 6-year data on maintaining that as a durable closure, which is just a wonderful result. So we have now conducted 2 studies. You saw the results from our 50-patient study, at least 5 of the sample patients, and we will be expanding that into now a further study under more control. To look at those results. Parkinson's is, of course, a very interesting area. This is a neurodegenerative disease. We've only treated 2 patients at this point, but we got strong positive results from both of those patients. And we are now expanding that in further clinical trials, small number trials for safety and initial, what we call feasibility or initial efficacy.
You mentioned COVID also as a development program. Do you have any insights there as to what that might be doing?
COVID is a very interesting area, also an interest for us. It's what we're specifically interested in is what we call long COVID. So these are patients that have been through the COVID process or initial infection, and they are compromised. They have pulmonary compromise, what we call long COVID or secondary COVID. And it's a long period of recovery, or in some instances, no recovery after recovery from the initial infection. And stromal vascular fraction is just such a powerful tool for inflammatory fibrotic responses. And we've been approached by some groups to look at response to COVID. And so we've initiated an effort with the FDA, and we will have an IDE in place very soon for investigating a number of patients with what's called long COVID. There is no therapeutic anything available at this point, and if we're able to show some level of response, I mean, this would be a very simple and very powerful way to address this a terrible situation in these patients.
Could you touch on reimbursement? Do you have any codes that you've secured, or is that a long game issue at this point still?
Reimbursement is always a long game issue, although we have a plan in place. We are certainly moving towards the full reimbursement, primarily first and foremost in osteoarthritis of the knee. We've established CPT codes, which are called at this point, because it's not fully FDA approved and blessed by CMS and AMA, what we have temporary codes. We've established temporary codes that will be used for further development of what in the end will be full reimbursement codes.
You touched on this also in your presentation regarding patents. Now, you use autologous cells from the patient. How do you protect patents, or how do you utilize patents to protect your technology using patient's own cells, for instance? How does that work?
We don't claim the patient's cells. That's theirs, and we can't. But what we do claim, we have apparatus and methods of use claims. So we protect the structure that is really, or the apparatus that really optimizes our ability to, at point of care, extract these cells in about 60 minutes with a very high efficiency, so that we get a point of care therapy. So we protect the mechanism and the process by which that happens. And then the method claims include methods of use of those apparatus and application as well.
Have you ever attempted to use cells that are not from the patient, but from other sources? And has that ever worked out in your estimation?
We have not. We do not use allogeneic cells, meaning cells from another patient. We are really focused in on maximizing the chances of a strong clinical result. Allogeneic cells, as you know, have a there is really a response, an immune response in the patient to anybody else's cells. I mean, that's the way the immune system works. That's the way it's intended to work. And if it doesn't work that way, there are significant issues. The technical other side of that equation is to create what's called an immunoprivileged cell, which is taking somebody else's cells and trying to essentially strip out the responses to the immune system. And there are still significant questions as to how long and how clinically effective something like that would be, and certainly safety issues. And the FDA, of course, is very concerned and very involved in the allogeneic cells. So we really try to optimize choosing if we can get the right number of cells. I mean, really, that's a question of cell availability. If you have enough of the best cells, which are yours, you would not need to find cells from somebody else. And so that's really what drives that whole allogeneic approach. And so we concentrate on enabling, with both technology and process, the ability to get autologous cells and to use those as the primary source for therapy.
I wish to thank Dr. Cimino for participating in our Capital Markets Investor Forum in conjunction with the World Stem Cell Summit being held. Thank you for your time, your excellent presentation. We enjoyed it immensely and hope to see more of it in the future.
Thank you for joining us for this C-Suite interview presentation brought to you by Channel Check. Visit our YouTube channel for more interview content, as well as virtual roadshows and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelcheck.com or click the link in the description to access equity research, news, and advanced market data on this and the 6,000 other small and micro-cap companies listed.